Hi, this is Shauna Gunaratney, and we will be continuing with the microbiology and pathophysiology of tuberculosis. So, Mycobacteria tuberculosis is an obligate aerobic bacillus, and its characteristic feature is a lipid-rich cell wall filled with mycolic acids, which you can see here. And due to this lipid-rich wall, gram staining is actually not helpful in diagnosing Mycobacterium tuberculosis or picking it up because the lipid-rich wall allows it to be impermeable to the gram stain, so it may not be gram positive or gram negative, it is gram invisible. As a result, we use something called the acid-fast stain, which I'll talk about on the next slide. So this mycolic acid-rich wall allows it to be acid fast, so it is stained, and then when using acid to decolorize it, it actually retains the stain and then is a different color than the surrounding tissue. So in terms of the microbiology, again, we use this acid-fast stain or the Zeal-Nielsen technique. So basically, the tissue is stained red with a red dye, and then an acid is used to decolorize or basically remove the stain from the tissue. However, this mycolic wall is able to resist the acid and holds on to that red stain. So it remains red, the rest of the tissue releases the red stain, and then the rest of the tissue is stained blue. So then the mycolic acids, the mycobacteria don't pick that up. And so they result as red among a background of blue, which is what you can see here. You can see the red mycobacteria as shown by the arrows. We also use a fluorochrome stain or fluorescent dye. The mycobacteria pick up this yellow-orange fluorescent dye and show up as fluorescent under a fluorescent microscope. So the structure of the mycobacteria leads to its pathogenic properties. So the peptidoglycan and the mycolic acids in the wall cause granuloma formation, and the phospholipids cause caseous necrosis, which is pathognomonic for tuberculosis. So this is sort of, you can see transmission here, and then as someone progresses through the stages of TB. So in terms of transmission, Mycobacterium tuberculosis travels via aerosol droplets. So these are under 25 micrometers. And these are released when someone coughs or speaks. Then they enter the respiratory tract in the human host. They typically land in the lower part of the upper lobes and the upper part of the lower lobes where the airflow is higher. They go to the alveoli and they are initially ingested by macrophages. They become phagocytosed and end up in the phagosome in this phagosome-lysosome complex. If the innate immune response, these macrophages in the phagosome-lysosome complex, is successful in containing the infection and destroying the mycobacteria, then the infection is cleared and someone's PPD, which relies on the T cell response, remains negative. However, it's unusual for the innate immune response to be able to control the infection. The mycobacteria escape the phagosome-lysosome complex, disrupt the macrophage, and then cytokines are released by the macrophage, which then recruits monocytes and dendritic cells to the area. Mycobacteria continue to multiply, and these monocytes that have been recruited develop into macrophages, but at this stage, there's very little inflammation. So you can see a schematic here of an alveolus with the mycobacteria in the phagosome-lysosome complex. They have escaped and disrupted this macrophage, and as a result, monocytes and dendritic sites are being recruited to the area. And then on the right-hand panel, you can see the granuloma formation when the T cell response is activated, and we'll talk about that later. So the mycobacteria have virulence factors that lead to its pathogenicity. So mycobacteria can delay the maturation of the phagosome, which again is instrumental in controlling it in the initial response. It also has urease, which neutralizes acid in the phagosome, so the human bactericidal enzymes that we have are less effective in destroying it. It also has antioxidants, which neutralized reactive oxygen species from the phagocytes that tried to control the infection. Mycobacteria also try to evade the immune response by staying in the phagosome so as to avoid T cell activation in the beginning. It also has these antioxidants that prevent early immune responses that are needed for antigen presentation. Again, another way that it's avoiding the immune system. And it also has something called cording, where it forms these serpentine long cords, which make it difficult for the immune system to control the infection. So if the infection is not able to be controlled, we move on to the next phase, the T cell phase. So those dendritic cells that were recruited to the area present antigens to the T cells, and then T cell immunity develops 2 to 3 weeks after infection. These T cells multiply, they activate more macrophages within this granuloma by secreting interferon gamma, which then recruits these macrophages. A granuloma is a collection of mature macrophages that arise in response to a persistent stimulus. These granulomas form with macrophages and lymphocytes, and they eventually contain the spread of the infection and sort of are walled off. Caseating necrosis occurs within the center of the granuloma, which inhibits mycobacterial growth. So here you can see a schematic of the macrophage and dendritic cell releasing a lot of cytokines and then eventually progressing to granuloma formation. So you can see here the dendritic cell or antigen presenting cell is secreting TNF alpha and then activating the T cells. And then the T cells are secreting interferon gamma, which recruit the antigen presenting cell and macrophages. So TNF alpha and interferon gamma are two cytokines that are very important in the T cell response in controlling TB infection. So when we look at the granuloma, it has 3 layers. So at the very center, there are central macrophages and giant cells within the bacilli. Then in the second layer, there are epithelioid cells, epithelioid macrophage cells, and at the very periphery, you see peripheral fibroblasts, T and B cells, and monocytes. So the caseating necrosis is found at the center. It is called caseating because it's named after casein, which is a protein found in cheese. So when looking at this under the microscope, the center of the granuloma looks cheesy, and that's why it's called caseating necrosis. So when we look at the tissue under the microscope, this is a slide showing the histology of the granuloma. So at the very center, the sort of darker pink, you can see the caseating necrosis there. And then the second layer are those epithelioid cells. And at the very periphery with those purple cells, those are the fibroblasts, T cells, and B cells. So if the granuloma is not able to wall off the infection, it may burst into a bronchus and form a cavity, and then the mycobacteria spread through the lymphatic pathway to regional lymph nodes, and that's why lymph nodes are actually the most common site of extrapulmonary TB. If it reaches the bloodstream, it disseminates to multiple organs in miliary TB, which is named for millet seeds based on the classic appearance and chest imaging.